I'm going to talk about African swine fever. And African swine fever, let me see if I can work with these. African swine fever is a very dangerous and threatening disease of pigs, domestic pigs namely, and it's shown here on these nice sculpture from a Spanish sculpture, you know, that this is really a global threat for the pig or swine husbandry, and now complicated with epidemiological data in Europe. So this disease is caused by a complex virus and is characterized by very complex epidemiological features, and its ASF-host interactions are complex also and not totally understood, and the vaccine and treatment are not available. And ASF prevention and control is based or are based on the implementation of strict sanitary measures. And this disease causes drastic socioeconomic impact at local, regional, and global scale. So that is the main. Those are the main characteristics of this disease. The disease is caused by African swine fever virus, and this African swine fever virus is the only member of the Aspaviridae family, genus Aspavirus, and is classified as a large nucleocytoplasmic DNA virus. And this is a big virus with about 200 nanometers diameter, a complex virus with an external envelope that we'll talk a little bit later, you know, with a capsid and an internal membrane. And this virus has a very large DNA, double-stranded DNA that codifies for more than 100 polypeptides or proteins, and these proteins are really in charge of different mechanisms. You know, they are structural proteins, around 50, from 12 to 15, 150 kilodalton, and proteins involved in the metabolism of nucleic acids, DNA replication, repair, and recombination, and mRNA transcription and processing, and enzymes involved in protein modification, some proteins that are associated with viral membranes and secreted, and mainly, you know, some proteins that interact or confuse the immune response of the host immune response, you know, like proteins proteins homolog to CD2 and AKB, and proteins that are involved also in apoptosis, like proteins homolog to YAP and BCL2, and with the redox metabolism. And there are also several proteins coded by those multigenic families, and many of them are not known what are their function. And this is a problem with this virus. ASF virus preferentially replicates on P macrophages, monocytes macrophages. And these pictures are pictures and images from 1975 from my colleagues in Portugal that they have studied deeply the replication in target cells in vitro. And as you can see, you know, the virus replicates mainly in cytoplasmic factories, inclusion bodies, and you know, it replicates. And when the virus comes out of the cells, the virus come out, you know, a little protected, covered with membranes of the macrophages themselves. And probably this is one of major points in terms of the host immune response, in which, you know, the animal is not able to produce neutralizing antibodies, as we know. And but recently in my lab, you know, my colleagues and a very nice lady, Margarida, has recently shown, you know, that the virus, the DNA replicates in the nucleus of target cells, both on macrophages and virus cells, and not only replicates but inhibits, you know, the DNA damage response mechanisms, and induces heterochromatinization by changing, you know, host epigenetic markers and disrupting subnuclear domains. So, in fact, there is an effect. I mean, previously to the formation of the new particles in the cytoplasm, the virus, the DNA of the virus does a lot of things in the nucleus of the cell, which has, you know, a lot of impact in cellular mechanisms, defense mechanisms. The virus, the natural hosts for the virus are all the members of the Swedish family, the domestic swine and the Suskroffa or the wild boar in Europe. And in Africa, there are other members of the Swede family, like the Fekoceros, the Potamoceros, and the Oloceros. Those swine, those pigs, are also sensitive to infection, but they have, infection does not progress in these animals and they do not show disease, but they can play an important role in epidemiology. Interestingly, this virus also, this is an arbovirus. This is probably, or is only the only DNA arbovirus known, and the virus replicates in Ornithodoros erraticus in Europe, when we had African swine fever since 1960 up to early 1990s, 94, 95. These vector played a particular role in the epidemiology of the disease. In contrast to Ornithodoros mobata in Africa, Ornithodoros erraticus does not allow the virus is not transmitted from the tick to the eggs. Only by the virus is transmitted in transstagial evolution steps of the tick. That's why, fortunately, we don't have any more ticks infected with African swine fever in Portugal and in Spain, certainly. And then Ornithodoros mobata, really, the virus replicates in these ticks, soft ticks, and it goes from the tick to the egg, and from the egg to the different stages of evolution. So, this is more or less, like a summary of the epidemiological actors for African swine fever. The domestic pigs get infected by oral nasal route, and what happens is that, and depending on the viral isolates, African swine fever is not only one virus. African swine fever are very many viral isolates from different areas, and they are of different virulence and related to virulence, related to viral doses, related to the host sensitivity. When the animals became infected, these virus replicates mainly in the lymph nodes, and then it progresses to the spleen and to the blood, but I would classify that two major forms of disease occur. The fatal with hyperacute, acute, and subacute forms of the disease, and the non-fatal, which means that many animals are chronically infected or non-apparently infected. And these animals are potential carriers, as well as the ones that survive the acute or subacute infections. The acute or subacute infections, they are, the clinical signs are similar to other diseases, namely to classical swine fever and to septicemic diseases caused by bacteria. But so, the animals have these listness, they have skin erythema or cyanosis, as you can see here, and they have unsteady movements. Appetite may be present at late stages of the disease. There is an increased pulse and respiratory rate, and sometimes, and very often, vomiting and diarrhea, sometimes bloody diarrhea occurs, and also with high discharges. And death occurs at different times depending on the violence degree of the agent. And mortality may approach 100%, or in acute phase, in acute forms of disease, or between 30 to 70% in other more moderate virus. And this disease, that somebody considered the disease is highly contagious, is not really highly contagious. It is contagious, but when it comes to pig unit, it starts like one here and one there and and one here, and one there, and in one week later, many animals become infected. This is not a disease that is transmitted by the air. This is not a disease that is transmitted like foot and mouth disease. And the lesions are very variable, and they relate to the different clinical signs, clinical forms. And so these are, examples of the most important disease lesions in acute and subacute forms of the disease, and they are mainly, congestion, edema, hemorrhage, and of all organs and tissues, and mainly in the lymph organs. So here you have, an aspect of a pig that had acute African swine fever. These are the tonsils, that are, hemorrhagic. This is hemorrhagic contents, stomach hemorrhagic contents that happens. Ulcers happen very often in pigs, and certainly, the spleen is enlarged like 3 times the size of spleen, and it's very congestive and even hemorrhagic. And of course, all the lymph nodes are congestive or hemorrhagic. And then there is another form of disease, which is the chronic form of African swine fever. And this chronic form that I'm going to show you appeared in Portugal in the early 60s. And because we in Portugal, we produced a vaccine by attenuation through passage on pig leukocytes, and by 150 passages in pig leukocytes. And that vaccine was applied in the field, and 500,000, 500,000 pigs had to be slaughtered after that, because there was a new form of disease, this chronic form of disease with loss of weight, irregular peaks of temperature, respiratory signs, skin ulcers, arthritis, and necrosis. Mainly those are the main lesions. And so this is a slow disease and a very low mortality that is very often complicated with secondary infections. And those are the original pictures of the lesions caused by this virus that has been adapted to pig macrophages. And at the time, that was, I'm talking about 1960s, and there was the way to attenuate virus, to pass the virus through cell cultures. And when this virus was tested in the lab, apparently, the animals could survive challenge with the original virus. But then when they were put in nature, in contact with African swine fever going on in the field, and so this is what happened. All these necrosis, skin necrosis, joints necrosis, and necrosis of the lung. And what happened now is that, in Europe, as you know, we have another actor that is a victim of African swine fever, that is the wild boar. And these pictures were kindly supplied to me by our friend Sandra Bloom in FLI in Germany. And in fact, the experiments and the studies done there, conducted there, have shown, that, when these wild boars have been inoculated with a highly virulent Armenian strain, so the strain that really attacked Europe. So there's, the animals have developed identical clinical signs and lesions to what happened to the domestic pig. And these are the original pictures from Sandra Bloom. The spleen, the lymph node, the gastrohepatic lymph node hemorrhagic, and hemorrhages in other lymph nodes, and certainly also hemorrhagic lesions in the kidney. So what is the problem now in terms of African swine fever, and what's happening in Europe? What's happening in Europe, it's really very, very scary. A few years ago, this slide would only show, the domestic pig, but how is the disease transmitted between animals? The disease is transmitted directly through contact with all the secretions and excretions from infected animals. And both species, the wild boar and the domestic pig, all, they are able to transmit the disease through blood, exudates, conjunctiva exudates, nasal discharges, saliva, feces, urine, genital discharges, and semen. So all these products are highly virulent, and the blood is highly virulent. One drop of infected or contaminated or virulent blood in one pig unit will be able to start an epidemic in the area and to conduct probably to 100% of mortality. And the indirect transmission is really very important in terms of the epidemiology of the disease. So, all those products can be, transmitted, can be taken. African swine fever does not spread by the air. African swine fever has to be taken. Somehow, African swine fever has to be taken by people, by vets, by, in food that is contaminated. And that is mostly, one of the major factors for transmission of the disease is the use of, swill feeding or waste that is contaminated. And that's how the disease appeared in Europe in the early 60s. And then, for instance, all the pork products, the sausages and hams, beautiful sausages and hams that we had in Portugal and Spain in the 60s, they would keep the virus for about 6 months. And those products could easily be transported to one place to another. And certainly animals that may come from infected areas that can transport the virus, and the vehicles in two ways. Vehicles that transport inside pork or pigs or pork products, or the vehicles that come from infected areas and they bring the virus in the wheels and outside, etc. So all these are risk factors for the introduction of the disease, and certainly the planes and the vessels, they are certainly vehicles that have been important in the dispersion of African swine fever in the world. So this disease was first found in Kenya in 1921 by Montgomery, and in fact he was the first one who tried to obtain a vaccine. And after the discovery of this disease was really very similar to the classical swine fever, and initially it was called, the new form of classical swine fever. And so this disease in 1957 came from Angola and to Portugal, near the airport. At the airport there were a couple of pig farms that were fed with leftovers from the planes. From 1957, then this outbreak disappeared, and around 1959, 1960, there is a new virus coming from Angola, from the west coast, and then it spread to different countries in Europe, and it went in 1970 to Caribbean countries, Dominican Republic, Cuba, and Haiti, and Brazil, 1978. And in Europe, the disease was completely controlled in all the countries except in Sardinia, in the island of Sardinia in Italy, that is infected since 1978. But in 2007, there was something that really occurred, and the virus has been classified using genotyping for the C-terminal of the P72 gene. And it is known, that the virus that came from West Africa and to Europe at the time in the 60s, belongs to genotype 1. But in Africa, there are 21 genotypes, and probably more than 21 at this moment. 21 genotypes relating to this P72 protein, or the gene that codifies the P72 protein. And this is probably due to the epidemiology of African swine fever in Africa, in Eastern Africa, that includes not only pigs, wild pigs, domestic pigs, but also the tick. And so in 2007, In 2007, the virus went from the east coast of Africa to Georgia in the Poti Harbor, and it spread to Armenia, Azerbaijan, and in 2007 to Russia. And that was the beginning. That was the beginning of what we are now facing, this problem in Europe. So in 2007, 5 wild boars were found in the south within the border. And from there, the disease has spread from 2007 to 2013, spread mainly in Russian Federation in two main areas, in the south and in the north and Tver area, and it went up to St. Petersburg, which is not very far from Finland. Okay, and so that was the situation in 2013, and 2014 the disease appeared in European Union, and so it was declared in Lithuania, Poland, Latvia, and Estonia. And these countries are now infected with African swine fever, and not only infected with a huge problem because wild boar is playing really a nasty role in the transmission and maintenance of the disease in these countries. And this is what happened in 2017. We have, as you can see, all these green dots mean wild boar infection, and we have also in Poland that at the beginning in 2014, African swine fever was mainly related to wild boar, is now advancing to domestic pigs. And certainly in last year in September 2016, there was an outbreak in Moldova. So the disease is really progressing in Europe, and the ways that this disease is progressing are the regular ones. I would say, the disease is maintained between the domestic pig population. At the beginning, the main problem was concerning the domestic pig population. But in these countries there are a lot of backyard farms, and these backyard farms they are in contact with the wild boars, and the wild boars became sick, infected with African swine fever. And now 95% of the cases reported to Office International des Épizooties, those 95% of the cases are from the wild boar. So this means, that the disease is in the wild, and so that means that there are many problems around to control the spread of African swine fever. And this is the current situation. So we have Russia that is considered infected with African swine fever. And it's funny because, Russia had first African swine fever, and then we were seconds, like in Poland, Lithuania, Estonia, and Latvia. And Russia just stopped all the importation from all the animal goods and products from Europe, and that means a lot of money that we are losing these days. And of course, the disease is endemic in Africa, in countries, in sub-Saharan countries, which is extremely important in terms of the danger of dissemination of the disease. So the prevention and control of African swine fever, it's a major question, a major problem. This disease is a major threat to the global farming sector and to the international trade of animals and animal products. At present, there is no treatment or vaccine available, and control of the disease is based on its detection and to be confirmed by rapid and efficient laboratory diagnosis. Fortunately, laboratory diagnosis of African swine fever is very efficient, and it's currently used and applied in Europe in many laboratories and reference laboratories, Unfortunately, not fortunately, is due to research efforts. Diagnosis of African swine fever is not difficult, laboratory diagnosis, but the main problem here is the early detection of the disease in order that, diagnosis can be provided, and if diagnosis is provided, sanitary measures of controlling the disease can be implemented. And these sanitary measures all know who are those sanitary measures because they are internationally organized. So, but unfortunately, and we see that in Europe these days, prevention and control measures are not enough to eradicate or to control the disease. Because unfortunately the disease is progressing, and this progress, in Europe has to do with many social, economic situations, and with political issues, and certainly economical issues. So, as the disease is in wild boar, and it is really difficult to control it under the current situation. So, what is necessary to do is to develop, efficient and safe vaccines is really very urgent. It's a very urgent need to sustain the advance of African swine fever. Europe is really worried with this problem, and vaccine is possible. Vaccine work is ongoing these days, and there are, basic data that really provides, the background for the vaccine development. The pigs that survive natural disease are protected against challenge inoculation with homologous isolates. Pigs surviving experimental infection with attenuated African swine fever resist challenge inoculation with homologous and heterologous isolates. That has been shown. Is in natural and experimental infections, viremia, and this is a problem, persists with circulating and ISF-specific antibodies, which means that really no neutralization occurs in normal conditions, although some neutralization has been shown in experimental conditions. And so vaccines that are based on the use of a live attenuated chemical and heat inactivated and genetically manipulated African swine fever have not proven so far to be efficient and safe to induce protection against different viral isolates. But ongoing strategies for vaccine development really open new insights to the obtention of a specific vaccine and safe vaccine. So, and this has been done through different strategies: durational gene deletion to produce attenuated ASF vaccine strains, subunit vaccines, DNA vaccines, protein and DNA vaccines, recombinant virus expressing ASF proteins, and replication and disc virus also have been currently studied to provide a basis for vaccine development. Many groups are dedicating to vaccine development, and it's hopefully expected that, not probably as quickly as we would like to, but I think that we have the basics to go ahead and to produce these vaccines. So in conclusion, I'd say that African swine fever is caused by a very complex virus that triggers an array of disease signs in swine, and this disease can easily spread and has devastating socioeconomic consequences in the countries where it occurs, causing significant impact on people's livelihoods, and this is very important, and on national and international trade of animals and animal products. No vaccine or treatment is available, and current re-emergency of African swine fever in Europe and the persistence of the disease in Africa are a threat to the global farming sector. There is no doubt about that. Besides the global efforts, I believe, beside the global efforts to sustain the advance of the disease through the correct implementation of prevention and control measures that are accepted internationally, I think that, countries and the scientific community should work together to obtain vaccine against this disease. And I'd like to acknowledge, people, the group in the vet school in Lisbon, and I'd like to acknowledge all the work that has been developed under the ESF Force project that was finished recently. It involved 18 institutions in Europe and Russia, and a lot of progress has been done under this project, and I thank you for your attention.